All right. All right. I see Ray is with us. Good morning, Ray.
Good morning.
You guys beat Glenville State, correct?
Yes, sir.
I think the only loss we had was the soccer team this weekend, right? I know the volleyball team had another sweep. We did good at State this weekend.
James got hurt yesterday.
Oh, really? Who are they playing against?
Tampa.
Who are they playing against?
The Buccaneers.
Oh.
We beat them.
Oh, you guys.
Yeah. James Winston, he played for us now.
Oh, see, I'm so out of it.
Yeah, he got hurt.
See, when I think of Jameis, I still think of
Florida State.
Oh yeah, but the Buccaneers. I did not know he left the Buccaneers.
Yeah.
Good morning, Hill.
Good morning.
I just don't have time for stuff I used to enjoy. These kids are wearing me out, man. My kids. All right, let's take a look here. What's it doing? Oh yeah. Get this. Get rid of this. PowerPoint. We were there. Where did we leave off? I think we got to the overview, and then we were about to start. Yeah, we left off when talking about the genetic, like GMO stuff, right? The glowing pigs, the glowing trees. Yeah, that's really interesting. I wish we had more time. I'd like to at least talk about GMOs a little bit. Everyone's afraid of GMOs, and sometimes I get it. Like there's something called BT corn. They take like a natural pesticide that doesn't affect animals, or that was the dumbest thing I've ever said. It doesn't affect humans, but it doesn't affect insects. So and it's from a bacteria. So they cut the genes out of that bacteria and they put it into the corn. So that way they don't have to spray pesticides on the corn because the corn just naturally kills it. But that scares people. And even though it's not scary, at least I get that, because then you're taking a poison, even though it's an insect poison, and you're putting it into the plant you're going to eat. So I see why that's scary. But then you have things, I think I've even talked about that this semester, things like golden rice. You take vitamin A that you get from carrots, which you need. You take that gene out of carrots and you put it into rice, because so many people in the world, that's all they live off of. So now you're giving people the better nutrition. And all you did was put it in something healthy. Or Arctic apples. You ever seen, what happens when you cut an apple and leave it out, right? It turns brown. That's because there's genes responsible for that. So for the Arctic apple, they just cut the genes out. That's it. There's nothing scary about that. I can see maybe you're getting scared of putting in a gene, but taking a gene out, that's not scary. Yeah. It's not locked. So nobody brought candy, huh?
I got you.
I'm just kidding anyway. All right, let's take attendance. Welcome to November. The last full month of the semester. We're getting there. For some of you, it's really, really crunch time, and I'm here to help. Anyway, let's get to it. Sullivan, not here. Hill is online. Byrne is not here. Tyrus is here in person. Lacey is online. Thornabar is here. Fitzpatrick is online. Hamlet is here. Huffman is not here. Johnson is online. Bayard is not here. Sigmund is not here. Pena is not here. Michael Baumgardner is here online. So is Ray. Hamrick and O'Brien are both here physically. Lloyd is online. Davis is not here. Elias is not here. Breeden is not here. And Cooper is here. Did I miss anybody? Everybody's accounted for? All right, we're going to jump back into it. Really quick with the reminders. Actually, you can ignore this. Again, the exam is tentatively planned for Friday. It's going to be online as usual. And as usual, if you want to take it in person, send me an email to let me know so I know to print one out for you. And as usual, it's only timed because our class is at 50 minutes long. So if you need more time, let me know. And we'll find a time to meet online or in person. As long as you're with me or I can see you and I know you're not taking, you're not looking at your book or notes or anything like that, then you can have more time. I don't care. So yeah, that's that. The lab, the moth lab that we did last week, that is due tomorrow. Just to remind you guys, I don't know how people keep missing this. I'm like, oh, my bad, I didn't know. It's been the same thing all semester. It's always due the Tuesday before the Wednesday. If that makes sense, the Tuesday before the Wednesday. Who'd have thunk it? Anyway, yes, last week's lab due tomorrow. This week, just to be clear, there is no pre-lab. Just to remind you, because I gave it to you so late, that's not fair for me to give it to you on Sunday and say, hey, make sure you have this pre-lab done by Tuesday. So no pre-lab. Obviously, the more you get done, the better, because when we come to lab physically on Wednesday, then you'll leave either at 1:50 p.m. or when you're done, whichever comes first. So obviously, the more you do before you get there, the better. Any questions about any of this? All right. Oh, yeah, and then for the exam, again, we'll do an exam review. It won't be during class time. We'll just find some other time. So, and if any of you have a particular time you would like to meet online, go ahead and start sending me those emails with your requests. Say, I'd prefer to do it this time because I'm here for you guys. So if everybody wants to do it at one time, I'll do my best to make sure my calendar is clear for that time frame. Any questions about anything? All right, let us get started. Oh yeah, for those of you online, I'm going to do what I've been doing. So you guys online will get the questions or the words. Excuse me, you will get the words. I'm going to give you some words, keywords for attendance. And then at the end, you'll just email me and give me those keywords. And obviously, if you're watching this as a video as opposed to live, you should have questions instead of words. But anyway, the first word for you guys is book. So the first word for attendance is book, like the textbook that you should be reading to prepare yourself for the exams. Here we go. Jumping back into it. Last thing we talked about was this whole idea of the genetic code and how it's universal. This is a really interesting topic, but I guess if you were to forget it for the rest of the semester, you'd be okay. It's not an important topic for the sake of understanding the other topics in this semester, but it is interesting. And again, if there was independent work, I would say look into it because it is really neat to see the different ways that we've swapped genes. Matter of fact, I'm going to share a documentary with you about some guy. There's this guy who used to be a molecular biologist, and he's kind of gone a little bit rogue, and he's doing his own experiments on himself. Anyway, let's talk about it. So to remind you, the first main bullet point we talked about last week was structure and function and replication. So you learned most of that was a review when you're learning about, DNA and RNA are polynucleotides, right? And the monomer that makes them up is nucleotides. And nucleotides are made up of sugars and phosphates and bases. And you learned all that. And that was a review. And then you learned that it gets replicated, as you already knew, in the S phase of interphase. And you also learned that the enzyme that does that is DNA polymerase. And I want to make sure I'm clear on that because I was watching one of my old videos and I don't remember if I even heard that word. So to be clear, the enzyme that does DNA replication is DNA polymerase. And it's in the name, right? Enzymes are named after their substrate or what they do. Well, that thing makes DNA. Anyway, then we got on to this whole process of how we go from DNA to RNA to protein. And when we go from DNA to RNA, that is transcription. When we go to RNA to protein, that is called translation. We already did the overview. So now we're about to jump into it and actually get into the details. So if you remember when we were talking about transcription and translation earlier, I said, no need to take notes right now if you don't want to, because soon we're going to go into details. And that is now. Now we're going to go into the details. So let's do it. So again, as you should already know, transcription, and I'm not going to ask you the definition of it. You just need to know, well, I'll get into that later. But transcription is the transfer of genetic information, right? We're going from DNA to RNA. I like how your book says transfer of information. We're not translating it. We're just transferring it. Just like the analogy I used last week. You've got this factory. In that factory, you've got a library that has these master copies of instructions on how to build stuff. And since that's your only copy, your master copy, you don't, like, whenever you need it, you don't just go and take the master copy out onto the factory room floor, right? You write it down. Plus, you don't need the whole book. You just need that one set of instructions for whatever it is you're trying to build. It's the same thing here, right? When your cells need to make a particular protein, they go and they copy that particular gene. Anyway, and that's what transcription is, DNA to RNA. Matter of fact, I'll underline that. If you like to make flashcards, you might like to abbreviate information, right? And just boil it down to a few words. In that case, you would do transcription is DNA to RNA. As we already know, the DNA strands separate when and where the process starts. Remember, we talked about how it's covalent bonds that keep each strand, right, a strand together. It's a covalent bond between the sugar and the phosphate groups. But it's a hydrogen bond that keeps the two strands together. Remember, we talked about how the hydrogen bonds are the weakest of the bonds. Therefore, it is relatively easy to take away those two strands, which exposes the bases. And that's where we get this bullet point right here. And again, this is a review. Each strand serves as a template. So the machine comes in, it reads it. It's like, oh, there's a C, so I'm going to bring in a G. There's a G, I'm going to bring in a C, so on and so forth. The RNA nucleotides take their place along the DNA template. You already know that. By forming hydrogen bonds. I have not specifically said that, but that should make sense because we know it's hydrogen bonds that keep them together. So it would make sense that when you bring in the RNA, that you're going to use hydrogen bonds as well. And that's how it works, right? So I'm going to show you some pictures of everything that's on the slide. But most importantly, that you need to know transcription is from DNA to RNA. And how does it work? Well, you need to know that the DNA bases serve as the template to say which RNA bases to bring in. I'm going to slow down. See note taken. I don't want to rush anybody. Are there any questions about this slide? So any idea why I would have a picture of a pin then on this slide? What in the world does that have to do with anything that's on this slide?
It's a Mount Blanc.
Is it?
Yep.
I don't know anything about fancy pens, but okay.
German made. That's the best writing utensil. That's the mark of a professional.
Oh, I did that on purpose, actually. No, I'm just kidding. I had no idea. So any idea then? Why? Like, what does a pen have anything to do with this slide?
Transcription?
Yeah, transcription. Remember, outside of biology, the word transcribe means to write something down. And that's literally what you're doing. Again, try to imagine my analogy of the factory, right? The library where you have all these master copies. You don't need the master copy. So you go in there, you just write it down. You're not translating anything. You're still going from English to English. At least in my analogy, you're going from English to English, right? So you're not translating anything. It's the same language, but you're just writing it down. You're going from a book to a piece of paper, or in this case, you're going from DNA to RNA. You're going from a full chromosome, right, to just a small little piece of RNA, because you only need that one gene. You don't need to copy the whole strand of DNA. So, as you already know, the nucleotides follow the same base pairing rules that you know. A goes with T, C goes with G. The only exception is, as you already know, we've said it so many times, there is no T with RNA, right? So you have a U instead. So when you get the question on the exam, which you will, where I say transcribe this, and I give you a whole strand of DNA, and it's a multiple choice exam, anything with a T in it is not the right answer. Because if it has a T in it, that means it's DNA. And if you're transcribing something, that means you're going from DNA to RNA. So if you're making a strand of RNA, you're not going to have a T. And here's the other important information, and this is really easy to remember, I hope. What machine is it that does all this magic? Well, it's an enzyme, and it's called RNA polymerase. So there you go. Those are the 2 enzymes you have to know for this entire chapter. I think the entire exam, we didn't really mention specific enzymes in the last 2 chapters, I don't think. So you need to know DNA polymerase is how we copy and make new DNA. And who to thunk it, RNA polymerase is how we make RNA. I like this picture. It's not from your book. You'll notice that this RNA polymerase in this picture is multiple colors. There's a little green spot here, some blue spot here, another blue spot here. This part's a different shade of green. And this is accurate. For your own knowledge, just so you know, DNA polymerase is not one molecule. It's a whole team of molecules. And this is part of how genetics can be so interesting and complicated, because you guys are getting the Cliff Notes version. It's really interesting how genes are turned on and turned off. Because to read a certain gene, whichever gene this thing is that we're looking at, to read it, you have to have all these different proteins recruited. And remember, proteins are made from genes. So to have that protein to read this gene, you had to have some other gene to encode to build that protein. And the process goes on and on. It gets so complicated. It's very fascinating, in my opinion. But again, for the exam, what you need to know on this slide is, again, RNA doesn't have a T. It has a U instead. And the machine, if you will, the enzyme that does the work is called RNA polymerase. And again, this is a picture not from your book. Let's look at a picture from your book. RNA polymerase doing its business. What is its business? Well, it's opening up the DNA strand so it can read the template, and then it just brings in the proper RNA nucleotide. So again here, I know this might be hard for you to see online, but right here there's a T, right, on the DNA, right? So the machine reads it, the RNA polymerase says, oh, there's a T, I better bring in an A. And then here's an A, so the RNA polymerase says, let me bring in a U. And then here's another A, so it says, let me bring in another U. And that's how it works. One by one, it just says, all right, next nucleotide. And the nucleotide hydrogen bonds here in the middle, right? That's what keeps the bases together. And what your book didn't mention, for your own knowledge, yes, it's the hydrogen bonds that first bring them together, but then the machine, the enzyme, does the covalent bonds. So this A will be covalently bonded to this A, and then that'll get covalently bonded to that U, and then that'll get covalently bonded to that U. But that's for your own information. Any questions about this picture? It's pretty simple stuff so far. Unfortunately, it does get a little bit more complicated. And by that, I mean you need to know the transcription itself is broken down into 3 stages. Initiation, elongation, and termination. You need to know them, and you need to know them in that order. Yes, technically it's called RNA elongation, not just elongation. But first of all, that should be common sense. I don't want to say common sense. It should be intuitive. What else are we going to be elongating, right? We're building an RNA strand, so it would make sense that we'd be elongating the RNA. And of course, initiation, yes, it's initiation of transcription, but yes, that makes sense because that's what we're talking about is transcription. And then there's termination of transcription. And again, yes, technically it's termination of transcription, but what else would we be talking about in this situation? So again, my recommendation for studying this, it's just a recommendation. Everybody's brain works differently, but for me, it works to memorize the big information and then start adding in details. So the first thing you should memorize from earlier is that we go from DNA to RNA to protein, right? That's the first thing to memorize. And then memorize that the process of going from DNA to RNA is called transcription. And then memorize the process of going from RNA to protein is called translation, right? And then, since we're talking about transcription, then memorize the fact that that's broken down into 3 stages: initiation, elongation, termination. And then once you have that memorized, then you'll memorize this other stuff I'm about to tell you, which is what happens in initiation, elongation, and termination. And the good news is, in my opinion, this is relatively, I won't say easy, intuitive. It's intuitive. Sort of like I was hoping mitosis would have been intuitive, like this phases of mitosis. These things that happen in prophase had to happen first. There was no other way around it. You had to get rid of the nuclear envelope. The thing that moves around the chromosomes has to attach to the chromosomes, right? All that stuff had to happen in that order. Same here. Plus, it's also in the name. This is a very well-named process. Here we go. The start transcribing signal is a DNA sequence called the promoter. You definitely don't need to write that down and memorize that verbatim. But the question on the exam is going to go something like, where on the DNA does transcription start? And the answer is the promoter. And that will be important because when we talk about translation, when we go from RNA to protein, we're also going to be talking about initiation, elongation, and termination. And you'll also be asked the question, where does it start? And for that process, has a different name. So remember, promoter is where transcription starts. And for your own knowledge, this is where the RNA polymerase, like I told you, and your book's not telling you, it's a bunch of different proteins. They all recognize the code on that promoter, like, oh, that's mine, right? That's where I need to be. And they fit perfectly, right? Because of the code. This is all chemistry going on here. But anyway, yeah, it's located at the beginning of the gene. And yes, that is where the polymerase attaches, which makes sense. That should be intuitive. Where does transcription start? It starts at the promoter. What happens at the promoter? Well, the enzyme that does the work, that's where it attaches. That should all be very intuitive. So basically then what you need to know is initiation, which is what we're talking about here, initiation of transcription. That is simply the attachment of the RNA polymerase to the promoter, right? That's it. That's when it starts. And you can see in this picture that's not from your book, at the bottom, and for you guys online, and this is a little bit hard, but here at the bottom left, that's the promoter, right? It's kind of like a lime green color. That's where it attaches. And you can see this purple part. That's the actual gene itself, is the purple part. But the promoter is right before it. That's where it attaches. Any questions so far? Pretty simple, right? And there you go. That's initiation. That's as complicated as it gets in this class. After initiation, as we already said, well, does anybody remember? It starts with an E. What comes after initiation? Good, elongation, because we're elongating this RNA strand that we're trying to build. Elongation. Nice and easy. Like I said, what happens in elongation? Well, the RNA strand elongates, or in other words, it grows longer. Technically what happens, as you can see in this picture, is as it gets longer, it peels away from the DNA strand. So you can see that happening right here, right in the bottom left corner of the DNA polymerase or RNA polymerase. You can see it peeling away. And then after that happens, DNA is done doing its business, it's done doing its job, it's done saying, all right, here's the template. So it comes back together and it's good to go. And this, I know we talked about in Chapter 8, Chapter 8, when we talked about, what was it? No, we talked about this Chapter 2. DNA duplication, right? Which happens in S phase of interphase. We're done talking about it. But I want to remind you, a lot of times that's where mutations happen. Because the machine that does that reads the code and then reads it wrong and brings in the wrong base. And that's where a mutation comes from. This is also another source of mutation, just so you know. I'm not going to ask you any questions about that. That'll be important later when we talk about evolution. This is another source of mutation. When mistakes happen here, you might get the wrong base. And that's what happened to the first person that ever had sickle cell, right? This enzyme read it the wrong way. It brought in one wrong, good grief, nucleotide, RNA nucleotide. It just so happened in that case, it didn't kill the person. And in that case, because of malaria, and we'll talk about that next chapter, having sickle cell was actually an advantage. Therefore, more people ended up having it. But we'll come back to that later. Again, for the exam, what you need to know is the second phase of transcription is called elongation, and it's simply when the RNA grows longer. I'm not even going to ask you these details about the fact that it peels away from the DNA and then the DNA comes back together. It's just there. And obviously it has to peel away from the DNA because we know, like I've already told you, this is all happening in the nucleus. And then once you get the RNA, that has to leave the nucleus. Or in the analogy I'm using, all this copying is happening in the library of the main set of instructions, right? And once that copy is made, you leave the library and go out to the factory room floor and build whatever it is that you're going to build. So we're done with initiation. We're done with elongation. What is the third and final phase?
Termination.
Termination. Good. It's in the name. The third and final phase of transcription is called termination. Where does it end? Just like when we talked about initiation, we also asked where does it begin? The answer is promotion. Now we're asking where does it end? It ends at the terminator. Nice and easy. Termination happens at the terminator. That is the end of the gene. And when the polymerase gets to that, let me write this here. When the RNA polymerase, just to remind you, gets to that portion of the gene, it detaches. Like, all right, my job's done. And then everything goes its own way. The polymerase goes and does its own thing. The DNA is back to normal. The RNA goes on to do its next thing, which we'll talk about later because we're following the story of the RNA. And that's it. Very simple, right? Again, what you need to know for the exam. The third phase of transcription is called termination, and it happens at the terminator. So what do you think would happen if there was some sort of mutation, maybe in S phase of interphase, so DNA replication, right? So let's say this thing's getting ready. We're not even here yet, right? Let's say this chromosome duplicates itself because it's getting ready for mitosis. And in duplicating itself, it messes up this portion right here. So it messes up the terminator. And now the terminator is no longer terminator. It no longer gives the stop signal. So what would happen in that situation? What do you think? If there was some kind of mutation in the terminator that no longer told the RNA polymerase, all right, this is where you're done. Your job's done. You can detach. What would then happen? Don't overthink it.
Just keep going.
That's right. Perfect. It would just keep going, right? And I think that's a test question. Or excuse me, I think that's a study guide question. It might be a test question. What would happen if there was no terminator is the way they put it. But why would there just be no terminator? I'm telling you why there would be mutation. If there was a mutation and the terminator wasn't working, yes, the RNA polymerase would just keep going. What would that result in?
Might die.
Yeah, you might die. But more immediately, the quick answer is it'll just be a longer strand of RNA, right? Instead of having a nice little short strand of RNA, the only piece you need, it would just keep going and have a longer one. But anyway, any questions about termination? Let's bring all three together and look at a picture of all of transcription. Here we go. Up at the top, for those of you online, I know it's hard to see. For those of you, up at the top left, in this first portion right here, right, you can see this little green spot is the promoter. RNA polymerase attaches, and then it starts moving this way, right? But when it attaches, that is called initiation. As it moves forward, that is called elongation. This RNA strand gets longer and longer, and elongation. And then finally it hits right here, which is the terminator. In this case, it's colored red. And then everything separates, right? The DNA goes its own way, the RNA goes its own way, and the RNA polymerase goes its own way. And again, it's the RNA that we're going to focus on. We're going to follow the RNA, follow the story of the RNA. Any questions? Should be pretty simple. The next slide I'm going to show you is a video, a little animation that just shows everything I taught you, but animated. So if you download the PowerPoint, you can watch it. Hopefully I'll find some actually better videos and share those with you via Google Classroom. All right. So there's no questions about transcription then, right? All right. Now I'm going to make things a little bit more complicated. I kept saying that it was simply DNA to RNA and then RNA to protein, right? I kept saying that when we go to DNA to RNA, that's called transcription. And when we go to RNA to protein, that's called translation. And that is all still true. Except now there's a little bit of a caveat. So we're done technically with transcription, but this new RNA that we made, it's not quite ready to go into this process of translation. So we have to do some stuff to it. This kind of reminds me of what was that process we talked about earlier in the semester? Yeah, it kind of reminds me of cellular respiration. Because remember, we said we simply go from glycolysis to the citric acid cycle to the electron transport chain. And then you found out it wasn't quite that simple because the product of glycolysis, which was the pyruvate, that wasn't ready to go into citric acid cycle, right? So we had to lop off another carbon dioxide. We had to attach an enzyme to it, blah, 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 blah. So same thing here, right? You're learning that it is a little bit more complicated than I initially told you. So let's talk about this processing of eukaryotic RNA. First of all, does anybody remember what a prokaryote is versus a eukaryote? Or can someone tell me what a prokaryote or a eukaryote is?
One has a nucleus, one doesn't.
That is a correct statement. More specifically, actually, that's a very specific statement, and that is actually what I was getting at. But in a broader sense, yeah, eukaryotes are like us, the more complicated organisms, plants, animals, fungi. We're eukaryotes. We have organelles, including a nuclei. Prokaryotes are things that you would normally associate like bacteria, and they do not have organelles, meaning they do not have nuclei, which is relevant to this chapter. That is correct. In prokaryotes, you don't need to write this down because I'm not going to ask you anything about prokaryotes, but just giving you a little contrast here. If we were talking about prokaryotes, which we're not, then this would be the end of the story. There is no nucleus. There is no processing, right? As soon as that RNA is made for bacteria, the code's there, it jumps right into translation. Here, we're not quite ready yet. Here, we've got to make some adjustments. And here's a new word for you, and you need to know it. Messenger RNA. You need to know this. Seems simple enough right now, but as we go through this chapter, I'm going to introduce more RNAs to you, and you're going to need to know the difference between all of them. So make sure you know. And this is a really important one, more so this semester than most semesters, because what kind of vaccine are people getting for the virus that causes COVID-19? It's an mRNA vaccine. So this is how, what you're about to learn, well, after we talk about the modifications, that's what happens with your vaccine. They're giving you the mRNA, which is funny because remember, I shouldn't say remember, you've probably heard a lot of conspiracy theorists. And what I love about them, and I'm not being facetious here, is I do love that they think outside the box. They're not just accepting answers. I'll leave it at that. I like to say something positive. But here's the thing. Notice you now know that mRNA comes from reading the DNA, right? It goes from DNA to RNA. So they're just skipping the whole DNA process, right? There is no reason for your vaccine to do anything to your DNA, right? This is all the next step. Anyway, again, for the exam on the slide, the important thing you need to know is the mRNA that we're talking about, when you go from DNA to RNA, right? That's specifically called mRNA, not just any old RNA. It is the messenger, which makes sense because going back to my analogy, if you've got a library with the books of instructions, like your master copy, and you go in there and copy it, you're writing down a message, right, to the people on the factory room floor. So that is the messenger RNA. It's bringing the message that's going to be translated. Any questions about this slide? All right, let's talk about this modification. The first modification is it gets something called a cap and tail. As far as the exam is concerned, you just know that it gets a cap and tail. Now I'm going to tell you what the cap and tail does. I don't think I'll go that detailed in the 50-ish question exam because we don't have that many questions to get that detailed. But the cap and tail, we're just adding extra stuff, like a cap and a tail, right? We're adding extra nucleotides, and it's just to protect it. Because as you might imagine, your cells do not want mRNA from foreign objects, like from bad sources, like viruses, which we'll talk about later. So if your enzyme sees mRNA that shouldn't be in your cell, it attacks it. It destroys it. Therefore, your own RNA needs protection, and that's what the cap and tail is. Now, this picture is not from your book, which means it has details that you don't need to know. For example, the tail is actually called a poly-A tail, meaning it's just a bunch of A's. Like, remember A, T, C, and G, or in RNA, A, U, C, and G? It's just a bunch of A's. And it's also on the 3' end, which is something I've not taught you and you don't need to know about. Meanwhile, the cap is on the other end, at the 5' end, and again, you don't need to know that. All you need to know is that one of the ways that RNA is processed before it leaves the nucleus as it gets the cap and tail for protection. Actually, yeah, let's just leave it at that. That is the simplified version. Know in the back of your mind that it is actually a little bit more complicated than this. I'm just giving you the simplified version. So if you go and do your own research on the virus that causes 19 or the vaccine, this is not enough information for you to fully understand it. But instead of starting right here, you're going to start right here, right? You're getting this good background knowledge to where you can move forward. Somehow, I have a lot of Facebook scholars on my news feed. My friends don't even know this, but yet somehow they're experts on how the vaccine works. Anyway, any questions about this slide? Okay, so that's one form of processing. The other form, this one's one, in my opinion, where it gets really interesting, RNA splicing. And it is exactly what it sounds like. You're just splicing. And here's where some new information comes in. Some new information you need to know. RNA, or mRNA, it's not all useful information. Same with DNA, right? Because this mRNA was copied directly from the DNA, right? So there is something on the DNA and subsequently the copied version of it, which is the RNA, called introns and exons. And you need to know this. Introns are the so-called junk DNA when it's on the DNA. Obviously, it's not DNA anymore, but you might hear the term junk DNA because it doesn't say, remember, the whole purpose here is to say build a protein, right? So it says, all right, bring me this amino acid. Good. Now attach this amino acid to it. Good. Now attach this amino acid to it, right? That's the purpose of these genes. Now, in this case, what I'm telling you is that there's some parts that don't say bring me an amino acid. And for the purposes of our class, we can call it junk DNA because as far as we're concerned, it does nothing. If you look it up, it does serve some purposes, but not this purpose. So anyway, the so-called junk DNA, I'm going to put a J there. Those are called the introns. And the parts that we actually need, those are the exons. And you need to know the difference between the two. And I hate that whoever chose those words because it can be confusing. It sounds like, to me, it sounds like introns, you keep it. And exons, you get rid of, right? It almost sounds like that. But here's what it means. The N in introns, well, let me back that up. Let's go backwards. Exons, what that means, the X and the exons means this is the portion that is expressed. The rest of it, as far as we're concerned, is junk, right? So the exons are expressed. The introns are simply the parts in between the exons. That's where the N and the introns comes from. So any questions about that? And this picture is not from your book. Again, you can see this pre-mRNA. I don't even know if your book ever uses that term. But as you know, we are talking about the processing of eukaryotic RNA, right? I just said earlier that RNA, that mRNA, it's not ready to leave the nucleus yet, right? So technically what we call that before it leaves, before it's done with its processing, it's called pre-mRNA, but you don't need to know that. All right, any questions about this slide? Okay, the second word for those of you online for attendance is beach. Like the sandy place next to the ocean, beach. Seems that Myrtle Beach seems to be the popular beach amongst West Virginians. Any questions about the word of the day or the second word so far? Okay, let's move forward. We're still talking about RNA splicing. Yeah, I talked about that. Yeah, so this is everything I've already showed you. It's no big deal. It's pretty simple, right? We get rid of the introns because we don't need them. However, it gets a little bit more complicated than that too. Now this is a little bit dated. No, we're going to go with that. Humans have about 21,000 genes, right? Someone looked that up. I remembered someone did the paper, not a whole paper, but they did that for independent work. That was the question they answered. How many genes do we have? It's about 21,000. Obviously, this number changes as technology gets better and as people do studies, but it's about 21,000. However, we have many, many, many more polypeptides than we do genes. So how is that possible? Because remember, the polypeptides, they come from these instructions, right? We have 21 sets of instructions, say, build this polypeptide. So then the question is, how do we have more polypeptides than we do instructions for polypeptides? And the answer is, oops, I'm about to skip a slide. The answer is alternative splicing. And you do not need to know the details of it. I might not even ask you any questions, but for your own knowledge, here we go. Let's look at this gene, right? So here's an exon here, an exon here, an exon here, here, and here, right? There's 5 different exons, all of which give codes to say, bring in this amino acid, bring in that amino acid, right? It's instructions on how to build the polypeptide. And then, of course, here's an intron, here's another intron, right? The introns are in between the exons. Now, what I told you previously in the last slide was that we just get rid of all the introns. For the most part, that is true. But it gets a little bit more complicated. Not only do we do that, but sometimes we get rid of some of the exons. And let me give you an example. So this, on the very bottom left, if you're online, if you can see where I drew that circle, this right here, this is just straight up a regular old, regular way to do it, right? You knock out the introns. You've got exon 1, 2, 3, 4, and 5, just like it should be, just like it intuitively should be, right? And then you get protein A. However, you could also get protein B in the middle, in which case, I know it's hard to see, but you have exons 1, 2, 4, and 5. So in this case, for protein B, we said, let's skip out. Let's leave out exon 3. So it literally cuts it out and then puts it back together, and it brings exon 2 over to exon 4, right? It leaves out exon 3. And you can see that's a slight difference, right? Protein B right here has a slightly different shape than protein A. And yeah, it's only slight, but remember, a protein, it's based on its structure, right? So if you make a slight change in its structure, you make a slight change in its function. And then, of course, right here, we have this protein C, which is where we have exons 1, 2, 3, and 5. So in that case, we cut out number 4. And again, you get a completely different protein. So that's a very simplified version. Also keep in mind that when we do this in real life, there's a lot more than 5 exons, right? You're talking a lot more, depending on the gene. So any questions about alternative splicing or splicing in general or processing mRNA in general? Perfect. Let's talk about translation. Now, translation, this is when we go from RNA to protein. It's a lot more complicated than transcription, which is what we just finished talking about, which is DNA to RNA. So because of that, your book breaks it down. Before we even talk about how it works, we talk about the parts. Like, here's all the parts involved. Introduce you to that. And then once you know all the parts, then we talk about how the parts work. So here we go. Let's talk about the ingredients. Now, I'll remind you, in translation, which is what we're about to talk about, we want to take this messenger RNA, this information in the RNA, and we want to translate it into an actual 3-dimensional protein. Knowing that, before we even get into it, what is one main ingredient of what we're about to do here? If we're going to take this mRNA and read the code on it and make a protein, what's the first main ingredient? mRNA, obviously. If we're going to translate the mRNA, then mRNA is the first important ingredient. Now, granted, in the exam, I'm not going to ask you to rank them, so it doesn't matter. That's the first one. Just know that mRNA is one of the ingredients to this process. So there we go. We have the message that we need to translate. Well, what else do we need? We need the translator, right? The machine that translates the mRNA. And I'm going to tell you what that is later. So you don't even need to write that down yet. So we have the mRNA. We have the machine that's going to translate the mRNA. What else do we need? We need some energy, right? All this requires energy because what are we doing? We're putting molecules together. As you know, that takes energy because we're taking this amino acid and attach this amino acid to it. Then we're going to attach another amino acid to it, so on and so forth. So that requires energy. Where does the energy come from, according to your book? ATP. So know that for the exam too. This requires energy. And whenever your cells need energy, they directly get it from ATP. That's what we're teaching you in this version of biology. I put a little star on there because technically it does get energy from other sources, but we don't worry about that. So we need 2 other components. We've talked about the mRNA. We've talked about the machine It translates to mRNA. We talked about the fact that that machine needs energy. Now we need to talk about something called ribosomes and something called tRNA. So let's talk about that. First of all, let's talk about the ribosomes, or as I said earlier, the machine that does the translating, right? If we're going to translate something, then we need an interpreter. Just like if you were at a UN council, right? All these people from all over the world, they're speaking in their own language. Everybody's got headphones on because somewhere there's somebody interpreting it, right? Somebody's like interpreting Swahili into Spanish, right? That's what an interpreter is. It goes from one language to the next. And that's what we're doing. We're going from one language, which is the nucleic acid language, to a different language, which is the protein language, the language of amino acids, right? So you need an interpreter. Specifically, as you already know, this is a good review from what we've learned earlier. It is converting the 3-letter nucleic acid words called codons. Remember, you need to know that language, the nucleic acid language is in, there's words, and those words are called codons, and those codons have 3 letters, right? So the interpreter converts those letters, those words, into basically amino acid words, as you already learned. Remember, I showed you that wheel, and I said, all right, if you start with a C and a G and then an A, then you end up with this amino acid. And if you start with a U and a G, then you end up with this amino acid, so on and so forth. So here's the important part. Well, this is all important, but here's the new information. This bullet point right here. It is the transfer RNA, abbreviated tRNA, that is matching the amino acid to their codons that forms the polypeptide. That is what your book says. If I were you, I would write it down like this. To me, this is the easier way of remembering it. Transfer RNA is the thing that's transferring the amino acids that you need, because remember, that's what you're doing in this process. You're just putting a bunch of amino acids together. Transfer RNA is just the thing that's transferring the amino acids to where you need them, which is the ribosome. And it's in the name, right? Transfer RNA. It's transferring something. Unlike the messenger RNA, which is carrying the message. And you do need to know the difference between those two for the exam. And then a third one. Your book points out that tRNA carries out 2 functions. This should make sense if they're transferring an RNA. Just think of it like a semi driver, right? A truck driver. What do? They're not always driving around with a trailer back there, right? Sometimes they're just, what do they call that? A bobtail? Just a bobtail, right? Just the machine itself, right? Not the trailer, whatever. So just like that situation, here we go. The transfer RNA, the first thing it needs to do is pick up the appropriate amino acid. Just so you know, each tRNA has its own amino acid that it works with. So then there we go. It's got its amino acid, just like a truck driver might go and pick up his load or her load. And then it's got to go to the right place to take it, right? So a truck driver doesn't just get a load and choose where to go, right? They say, hey, take this and bring it right here. And that's what happens here. So the tRNA, it's picked up its load, and then it recognizes the appropriate codon in the mRNA. It says, all right, this is where I need to bring this amino acid. I picked up the amino acid, the specific amino acid that I carry, and I brought it to this specific codon. And it's structure. It's the structure of the tRNA that allows it to do this. And we're going to talk about that. But for the exam, the most important thing you need to know is that the tRNA is the thing that brings in the amino acids. And remember, that's what this whole process is about. It's just attaching one amino acid to the next. We're trying to make a chain of amino acids because that will turn into a polypeptide, which will then fold up and become a protein. And we'll talk about that later. So let's talk about this unique structure. I'm going to talk about it quickly because it's a little bit less important because I'm not going to go into detail with this on the exam. Transfer RNA is made up of a single RNA strand. I find it interesting that your book mentioned that because as far as we're concerned, all RNA is single-stranded. Remember, we said that was one of the main differences between RNA and DNA. One of those things that you have to know for the exam. So you might say, then why bring that up again? Yes, we know it's single-stranded. I think the reason your book points that out again is if you look at this picture of it, right? That's a picture of tRNA. Look at right here, this portion right here. That looks double-stranded, right? Because you have this G hydrogen bonded to this C, and this C hydrogen bonded to that G, so on and so forth. So it looks double-stranded. And I mentioned this earlier in the chapter. I said there is a point in this chapter where I'll talk about an RNA that folds up on itself, and it looks double-stranded. This is that point, right? So it's not double-stranded. It's still single-stranded. It just looks like that. But not that that really matters because we don't go into this kind of detail in the exam. Before I show you the next slide, the third word, for those of you who are online for attendance, is movie. Like the thing you would watch in a theater or on Netflix or HBO or whatever. Any questions about the other word? All right, let's move forward. We're still talking about tRNA. At one end of the tRNA is something called an anticodon. And as you might imagine, the anticodon reads the codon. That's the part that recognizes, hey, this is where I need to go. At the other end is where the amino acid attaches. So again, you can think of it as a semi. The back of the semi is where the amino acid attaches, right? That's where the cargo goes. The front end, that's the anticodon. You can almost think of that as the GPS in the cab of the truck. You put in the exact location that it needs to go, and that's how it knows where it needs to go. And then here's a picture of what we're talking about. I'm going quick because I'm not going to ask you about this. So here's what it actually looks like on the left, right? Here's the anticodon that reads it. So on this anticodon, if that was a CGC, it's looking for its base pair. It's looking for the opposite, right? So it's looking for a GCG, right? Anyway, that's how that works. And then this picture, I should have drawn an arrow right there. So the picture on the left is a more accurate representation of what it looks like. The picture on the right is a more useful representation of what it looks like. Because to me, that's kind of what we need to picture when we're picturing this process. This thing is just carrying stuff. And this top little purple spot right here, that's where the amino acid attaches, right? That's where the cargo goes. This bottom spot right here with the anticodon, that's where it recognizes where it needs to go. We don't really have time to get into ribosomes. Let's just end 3 minutes early. And the last word, for those of you online, is bucket. As in, like if you go to a beach, you might put some sand in the bucket. Or if you go to the movies, you might put some popcorn in a bucket. And again, for those of you who are online, you need to email me and say, hey, I'm writing for attendance on this date. Here are the keywords. And if for some reason you were pulled away online, but I still see you here, then if I post the questions before you email me, then you'll just have to do the questions instead. Any questions? All right, everybody. See you on Wednesday. Morning. Are you a fan of all Miami athletics or just a fan of the city? I forgot. I think I already asked you that before.